Welcome to this explainer. Okay, we have got something absolutely massive to talk about today. We are exploring a medical breakthrough in artificial intelligence that is literally rewriting the rules of human biology, survival, and drug development. For decades, you see, scientists have been locked in this high-stakes race against time, trying to understand the microscopic machinery that drives our bodies, and more importantly, what exactly goes wrong when we get sick. Well, AI is officially turning that centuries-old observational science into a high-speed predictive engine. We are looking at a total paradigm shift in how we treat disease.
5,027. Just let that number sink in for a second. For years, this number represented the ultimate biological cold case. These were the quote-unquote unsolvable human proteins. Now, proteins are the fundamental building blocks of life, right? They govern everything from how we digest our food to how our immune system fights off viruses. But here's the catch. To understand how a protein works, you have to know its exact three-dimensional shape. And for 5,027 critical human proteins, traditional science had basically hit a brick wall. We knew they were there, but we just couldn't see them.
So this massive, invisible gap in our knowledge became known in the scientific community as the dark proteome, which naturally begs the question, what exactly is hiding in the dark? Well, the answer is both terrifying and incredibly tantalizing. Locked away in these invisible, unmapped structures were the very secrets to human disease. We're talking about the mechanisms behind severe neurodegenerative conditions and the elusive pathways of cancer. But if we couldn't see their shapes, we couldn't design treatments to target them. It's really that simple.
Okay, let's dive into the actual numbers here, because they are staggering. Before AI, our structural coverage of the human proteome was agonizingly slow. It was stalled out at just 48% using traditional, painstaking methods like X-ray crystallography. But after AlphaFold 3, that coverage rocketed up to 76%. And remember those 5,000 plus unsolved mysteries? They plummeted down to just 29. No, seriously, 29. The AI absolutely shattered the traditional bottlenecks that have been holding researchers back for decades.
Now, to really grasp why this is such a game changer, we need to talk about structure-based drug discovery, or SBDD for short. This is the process of designing new therapeutic drugs based on the 3D structural information of biological targets. Think of a disease target as a highly complex microscopic lock, and the therapeutic drug as the key. If you can't see the exact shape of the lock, you're essentially just fumbling in the dark trying to carve a key that fits. Seeing the precise shape of a protein is literally a matter of life and death when treating complex diseases.
Enter AlphaFold 3, the ultimate AI detective. Developed by Google DeepMind, this system is the technological leap that finally cracked the molecular code. Now, while earlier versions were great at solving the single-chain protein folding problem, AlphaFold 3 went way further. It completely bypassed those human-only research bottlenecks to predict not just isolated proteins, but how they interact with the entire cellular environment. It's a fundamental, ground-up shift from human observation to pure machine precision.
So, how does this AI detective actually operate under the hood? Well, it's a massive architectural shift. First, it moved to a pairformer, model to look incredibly closely at how amino acids pair up with one another. Second, it utilizes a diffusion model. Conceptually, this is kind of like the AI that generates those digital art pieces you see online, but instead of predicting pixels, it's generating exact atomic coordinates. Finally, it maps interactions across a whole range of biomolecules. We're talking DNA, RNA, and ligands, the whole shebang. It's predicting these complex, multimolecular assemblies all at once.
Now, I want to be clear here. This isn't just some black box of magic. Mechanistic interpretability studies, specifically a framework called AlphaFold, reveal exactly how this AI thinks. It shows that the AI essentially acts as an incredibly advanced fold recognition system. To avoid hallucinating fake structures, it relies heavily on phylogenetic diversity. In plain English, it looks at deep evolutionary family trees across completely different species to anchor its predictions. It is literally relying on the deep history of evolution to accurately predict biology today.
And here is where it gets incredibly real. 70%. In a major milestone back in 2023, researchers used AlphaFold 3 to map previously unsolvable cancer structures. By doing so, they cut the initial chemical screening time by a massive 70%. That is absolutely huge. What used to take years of frustrating trial and error in a physical lab was compressed dramatically.
We are witnessing a profound transition right now. As you can see, molecular biology is moving from an observational science straight into a predictive, engineering-driven discipline.
We are no longer just looking through a microscope, documenting what we see and crossing our fingers hoping to find a match. We are engineering solutions rapidly and precisely, completely based on highly accurate algorithmic predictions. So, what does this actually look like in practice? Basically, high-throughput AI structural databases are compressing years of traditional drug design into literally minutes. By relying on these vast digital repositories, researchers can now run rapid virtual screening algorithms. The AI systems can simulate thousands of molecular interactions instantly. They pull the best, most viable candidates to the surface, completely ready for real-world testing.
And the best part? This incredible tool isn't locked away in some secretive corporate vault. The open-access AlphaFold database has already supported over 500,000 global research projects. By 2024, this single resource became a fully democratized open-source tool. It's actively empowering independent initiatives all over the world to accelerate their own life-saving medical research.
200 million. Thanks to that open access, the scientific community now has over 200 million mapped protein structures right at its fingertips. That is virtually every single known protein sequence on the planet mapped out and ready to use. This staggering volume of data is drastically lowering the cost of structural biology research and completely leveling the playing field for scientists globally.
So pulling this all together, why does this matter to us? Because it fundamentally alters the entire future of medicine. With this tech, we are accelerating toward personalized therapies designed specifically for your body, down to patient-level protein interactions. We're advancing synthetic biology, which allows us to design entirely novel proteins for new materials. And crucially, it supercharges global health and pandemic preparedness. It gives us the structural tools to rapidly understand and neutralize new infectious diseases the very moment they appear.
The dark proteome has been illuminated. The invisible is finally visible. I want to leave you with this final, provocative thought. Now that the building blocks of life are fully mapped out, what disease will humanity cure next? The microscopic locks have been identified, the speed of discovery has been permanently altered, and the next massive life-saving breakthrough might just be an algorithmic prediction away. Thanks for exploring this with me today. Keep analyzing the data and stay curious.